Right. Good afternoon, everyone. My name is Takashi Tsukamoto. I'm with the Johns Hopkins Therapeutic Core of the Johns Hopkins Center for the Advancement of HIV Neurotherapeutics. And it's my pleasure to introduce our speaker today, Dr. Luca Shaitaj at the University of Bristol in UK. As far as I know, this might be the first time we invite speakers from Europe. So it's almost a time for beer for him, but thanks for sticking around. Dr. Shaitaj obtained his PhD in infectious disease at the Italian Institute of Health in Rome, where he contributed to establishing a macaque model to study SIV persistence under antiretroviral therapy. And during his postdoctoral work at Heidelberg University Hospital in Germany, he investigated how redox and ion metabolism influence the development and maintenance of HIV latency. So in 2022, he established his own research group at the University of Bristol in the UK, and his lab focuses on the understanding how viral infections reshape cellular metabolism and on developing pharmacological strategies to target disease alternation, which is something that the therapeutic core of the Johns Hopkins is quite interested. So actually, if you Google his name, you will find a Wikipedia page dedicated to him, as he's a well-accomplished chess player with a grandmaster title. So I guess he's now transitioning this talent to deal with the biology of infectious disease, which is almost like playing a chess game, I guess. So the title of his talk today is Leveraging Redox Dysregulation to Target Latent HIV Infection. So I'd like now turn this over to Dr. Shaitaj.
Thank you very much, Takashi, and thank you all for joining me today. So I will just share my screen. Can you all see it?
Yes.
Excellent. So thanks again for the kind invitation, which gives me the opportunity to share some of the work that we've been conducting in the last few years. And really the goal of this work is to try to disentangle a bit this relationship between redox metabolism and HIV infection. So as mentioned in the kind introduction, I work at the University of Bristol, which is in Southwest England, and this is one of the landmarks. It used to be, I think, the longest suspension bridge in the world, and it's still quite a nice thing to see. So I assume most of you are not familiar with what my lab does, because it's a fairly recent lab, but basically we try to find how metabolic dysregulation can be induced by viral infections, and in particular by human retroviruses. So apart from HIV, we also do some work on HTLV1, and we have also done some drug repurposing on coronaviruses. But ultimately, the way we look at it is for finding it. Our goal is really to find markers of metabolic dysregulation that are druggable, that can be therapeutically targeted. So that's what we would like to do. So some of you, I assume, will be familiar with this, so apologies if it's a bit too basic, but I thought since a lot of the talk will be focused on redox metabolism, we could just quickly go through an introduction to explain what it is, so we're all on the same page. So redox simply means an exchange of electrons, where a species will get oxidized and lose electrons, and another species will get reduced and basically gain an electron. Now, if the species that is gaining an electron that is getting reduced is oxygen, this can lead to the development of so-called reactive oxygen species. And these are simply derivatives of oxygen that, as the name itself says, are quite unstable, quite reactive. And the reason why they're so unstable is that they have either an unpaired electron or, in any case, an unstable electron configuration, which makes them so reactive. And when we look at the cellular context, we can have different sources of reactive oxygen species. Some of them are exogenous, for example, radiation, or as we will see, infection. Some of them are endogenous, and most of the endogenous sources are really metabolic. One example is, for example, the electron transport chain, where by definition, the final acceptor of the electron chain is oxygen. So this can give rise to reactive oxygen species. But this can also happen in peroxisomes, in the ER, and enzymatic reactions. So overall, it's quite difficult to disentangle, also because they are so reactive and unstable. It's quite difficult to disentangle the source of reactive oxygen species. But what I hope my talk will provide support for is that we don't necessarily need to do that as long as we understand how cells are trying to cope with an imbalance in reactive oxygen species. So therapeutically, the way cells react to them is, I would argue, more important than exactly understanding where they come from. And the way cells react to them is really by using antioxidant defenses. And antioxidant defenses are based on the general mechanism of trying to basically correct this unpaired electron or electron imbalance, typically by providing a further electron, by donating a further electron to a free radical. And since the antioxidant function is important because reactive oxygen species are pretty much ubiquitous, there is a certain level of redundancy in antioxidant species. They can be classified as enzymatic and non-enzymatic, but the take-home message here is there is some redundancy. There is also some specialization. For example, superoxide dismutase is responsible for detoxifying superoxide, while catalase is responsible for detoxifying hydrogen peroxide. So there is both redundancy and specialization, but for the purpose of this talk, I'm really going to talk mostly about these two antioxidant pathways more than just antioxidant factors, glutathione and thyroidoxine. And the final piece of introduction that I would like you to keep in mind for the rest of the talk is that oxidants are part of normal cellular physiology. So when we basically consider reactive oxygen species, we consider them in terms of a balance in the cell. So if this balance is typical, then we have enough antioxidants to compensate for the level of oxidants. And what this brings to is simple normal cell physiology. So for example, neutrophils require reactive oxygen species to ensure effective immunity. Reactive oxygen species are also required for cell signaling, but also, for example, for post-translational modifications, bisulfite bonds in proteins, and so on. However, if the balance between oxidants and antioxidants is altered, and there are too many oxidants in the cell, then a situation, a condition called oxidative stress can ensure. And this is associated with, for example, cell death, DNA damage, or at the organism level. It can also be associated with neurodegeneration and aging. So it's really a balance between these factors. Now, moving on to HIV, the relationship between HIV and reactive oxygen species was initially investigated quite early in the HIV research, in the history of HIV research. The initial reports linking HIV infection with a burst in reactive oxygen species were from the early 90s, really. And it is also understood, based also on what I said in the last slide, that part of the pathogenesis that can be induced by HIV infection, in particular neurocognitive disorders, might be linked to persistent oxidative stress induced by HIV replication. Another idea that was developed early in the course of HIV research was to try to use antioxidants, such as, for example, glutathione, in order to reduce the replication of HIV. Now, as you may imagine, this idea was a bit more popular during the early stages of HIV research, since now we have very, very effective drugs able to selectively inhibit the replication of HIV. The use of only partially selective agents to do so is less appealing. However, the current condition with antiretroviral drugs has also led to a paradigm shift where instead of trying to inhibit the replication of the virus, there is the attempt of trying to reactivate the virus from latency and, if possible, also to kill persistently infected cells during antiretroviral therapy. So in order to investigate the regulation of reactive oxygen species, we initially used a relatively simple model. This is a post-activation infection model. So we isolated primary CD4 T cells, activated them by engaging with beads, the CD3 and CD28 receptors. And then we split the cells in two conditions. One of them was infected with HIV, the other was left uninfected, and we sampled different time points, having always a matched uninfected control in different time points. And we gave IL-2 to preserve the viability of the cells. Now, the model has some issues. One of them is definitely the different viability of the two conditions. However, it does mimic what we wanted to capture to some extent, the transition between a stage where HIV is actively transcribed and translated and productive infection pretty much, and a stage where HIV replication is lower or ceases entirely. And the reason why I'm saying that is because basically in the early time points, we can see both a robust protein HIV expression and mRNA expression. However, this expression goes down 2 weeks post-infection. However, during this time point that we dub latent infection for the purpose of this model, we still see we still see HIV DNA in the nucleus as seen by HIV DNA fish. And we also can detect HIV integrated DNA in the culture, and we can also induce further replication, basically P24 expression. I'm sorry.
That's okay. Actually, you froze for like last 30 seconds. So if you wind back to 30 seconds.
Okay, so sorry for that.
That's okay.
The point that I was trying to make is that we see an initial expression of HIV gag protein and transcript, and this expression goes down and basically almost undetectable levels. However, in the time point that we selected as our latent time point, we can still see HIV DNA in the nucleus of the cells as seen by DNA fish, and we can detect integrated DNA in the culture as detected by ALU-PCR. We can also, to some extent, induce a new production of P24 by reactivating the cells during this time point, pretty much, either with CD3, CD28 beads or with PHA. So the point that I'm trying to make is this model does capture some of the transition between active replication of the virus and the moment where this replication of the virus ceases or is reduced. So by using these different time points that were sampled in parallel for infected and uninfected cells, we measure the ROS as the first thing. And we used basically a fluorescent probe that by getting oxidized in the presence of ROS will emit a green fluorescence. And what we saw is that when we compared productively infected time points, meaning time points where we could detect robust expression of HIV, we had higher levels of reactive oxygen species in infected as compared to uninfected cells. However, in the latent time point, or at least in the time point that we determined as such, based on the fact that HIV was not as expressed, we did not see this difference in reactive oxygen species. Perhaps more interestingly, when we restricted the analysis to those cells that were positive for HIV DNA based on the fish DNA signal, then we saw that the cells positive for HIV DNA during our productive infection time points had higher levels of ROS as compared to the cells positive for HIV DNA in our latent or low expression time point. Now, one important thing to remember, as I said also in the introduction, is that when you have an excess of reactive oxygen species, you will have an antioxidant response trying to cope with that. And antioxidant factors, as we saw, are many. However, the response is highly coordinated by a transcription factor, a master transcription factor, which is called NRF2. Now, in basal conditions, NRF2 is in the cytoplasm, is bound to KEAP1, and it's also ubiquitinated and induced to degradation through the proteasome. In the presence of reactive oxygen species, this binding between KEAP1 and NRF2 is lost, and NRF2 is imported in the nucleus, leading to the transcription and then translation of antioxidant proteins. So one of the things that we wanted to see in our model was whether we could detect more NRF2 in the nucleus of infected cells in the early stages of infection, where we saw the burst of reactive oxygen species. And that was indeed the case. Again, rather interestingly, I would say, even when we considered only the infected cell cultures, so here we are comparing uninfected and infected cells, but here we're considering only an infected cell culture, those cells that were positive for HIV RNA fish, so those cells that are actively transcribing HIV at that moment, but also more expressing more highly NRF2. So we asked ourselves what would happen if we knock down NRF2, and we saw an increase with a temporary knockdown through short interfering RNAs. We saw an increase in HIV transcription. Moreover, during the stages where HIV expression goes down, if you remember, it's towards the second week, we saw first an increase when HIV replication goes up of antioxidant. These are downstream antioxidant genes that are modulated by NRF2. So basically, these are an indication that NRF2 is in the nucleus and is inducing the transcription of antioxidant genes. So this expression of antioxidant genes was increased in infected cells during the active stages of the replication, but it went down, at least in part, afterwards. And finally, if we induce reactivation of HIV, we saw the same antioxidant genes going up in the cells that are reactivated. So all in all, these lines of evidence suggest a relatively simple model where the expression of HIV will lead to reactive oxygen species generation, will lead to the activation of NRF2 and to the activation of the antioxidant response. Now, this antioxidant response will favor, or at least correlate with the shutdown in the expression of HIV. So if we knock down this antioxidant response, perhaps we can induce the reactivation of HIV from latency. Now, although the model is really simple in this iteration, I would say, it's not as simple to put in practice because we cannot obviously shut down the entire antioxidant response. There's a reason why it's there. So we have to select something that we can target and that is relevant for HIV latency. So this brings me to the question, which NRF2 targets should we inhibit? And in order to explain how we came up with a tentative answer for this question, I need to make a digression, a detour. This digression is an attempt to expand our view of metabolic imbalances in the context of HIV infection beyond reactive oxygen species. Now, one line of evidence that we saw in the same model that I described before is that the expression of the glycolytic pathway genes is progressively downregulated in infected cells after the initial infection. And this is really interesting because a couple of years before that, the group of Sirion in France has shown that cells, lymphocytes, T lymphocytes, characterized by higher glucose uptake, which means also higher glycolysis, tend to get more infected. So basically, in the early stage, if a cell has higher glycolysis, will tend to be more infected. However, our data are suggesting that in the later stages, downregulation of glycolysis tends toward latency. So to understand a bit better this relationship between lower glycolysis and latency, we reanalyzed a data set that was published by the group of Angela Ciuffi in 2018. So what the group of Angela Ciuffi did was to take CD4 cells. Again, it's a post-activation infection model, but in this case, they use an HIV GFP virus, which allows them to sort GFP positive cells and have a pure culture of infected GFP positive CD4 T cells. Now, by keeping these cells for long-term culture, they achieve the loss of GFP signal, which can, in part, be reactivated by giving, for example, by engaging the T cell receptor or with vorinostat, SAHA. Now, the authors published some nice experiments and results with this, but we were interested in their single-cell RNA-seq. And the reason why we were interested in that is that they identified two clusters of gene expression that characterized cells that were more likely to get reactivated or not from latency. And this is very important because, for example, the Siriciano lab has shown that not all cells, all latent cells, despite having intact HIV DNA, are going to get reactivated, even in the presence of very strong reactivation stimulation. And so we wanted to see whether the cluster 1 of gene expression would also correlate with lower expression of the glycolytic pathway. And this was indeed the case throughout all the conditions that the authors analyzed, suggesting really that this lower expression of the glycolytic genes was a hallmark of cells that were latent and also less likely to get reactivated. So we also wanted to expand a bit beyond lymphoid cells, beyond CD4 cells, because both our model and the model of Angela Ciuffi are based on CD4 cells. So we collaborated with the lab of Jonathan Karn, trying to see whether in other models of reservoir, we could detect the same pattern, meaning lower glycolysis and higher latency, favoring latency. And so we collaborated with the lab of Jonathan Karn, and particularly David Alvarez Carbonell, who had set up a model of immortalized microglia, which I assume many of you will be familiar with. And this model is based on the immortalization with the SV40 T cell antigen and reverse transcriptase of the endogenous human retrovirus. And it consists of clones that are uninfected, so the C20 is uninfected, or the clone HC69, which is infected but latently infected with the GFP HIV strain. And what is nice about this clone is that can get reactivated, the expression of HIV detected as GFP can be reactivated in the presence of TNF. So what we did there was first a metabolomic analysis where we saw basically that in reactivated cells, glycolysis was one of the most enriched pathways. But more interesting, we also saw that the latently infected clone microglia was characterized by a lower expression of NADH as compared to uninfected microglia. And this is important because NADH is one of the main products of glycolysis. So glycolysis leads to ATP, pyruvate, and NADH. Which also brings me to the end of this relatively long detour to try to answer the initial question. Which NRF2 targets should we actually target? Which antioxidant factors that are favored by NRF2 nuclear import should we try to target for our purpose of eliminating infected cells? Well, since we saw that glycolysis and NADH regulation is a hallmark of latent infection, and since cells are in any case reliant on reducing factors, we postulate, and we also have, we saw that these cells will be more reliant on NADPH, which is typically generated, but not only generated through the pentose phosphate pathway, which we saw was not downregulated in latently infected cells. And the reason why NADPH is so interesting is that it's the link between the function of thioredoxin reductase and glutathione reductase, which are both essential for the function of thioredoxin and glutathione, so the two antioxidants which are both modulated by NRF2. There is also another reason to prefer glutathione and thioredoxin as potential targets, and the reason is that there are good drugs to inhibit both. So for thioredoxin, in particular for thioredoxin reductase, there is an approved drug used for rheumatoid arthritis called Oranafin. For glutathione, there is a drug that has been tested in humans, although it's not approved for human use, and which inhibits a key enzyme, gamma-glutamyl cysteine synthase, which means that by giving buthionine sulfoxamine, BSO, the levels of glutathione will quickly, although reversibly, drop. Another thing that is interesting about these two pathways, glutathione and thioredoxin, is that they tend to be redundant. You remember that I discussed a bit that antioxidant systems have a certain level of redundancy. Well, if we inhibit both of them at the same time, we get a level of oxidative stress that is synergistically higher as compared with the use of each drug alone. So our hypothesis was obviously that latency could rely on these antioxidant systems. So we tried to see whether we could disrupt latency in a variety of different cellular models, cell lines and primary cells of lymphoid and myeloid origin, by using either drugs separately or both drugs in combination. And as expected, we saw that both drugs in combination work best, although at a different extent based on the cellular model that we were using. Moreover, we are inhibiting antioxidant pathways. In this case, we are inhibiting both thyroidoxine and glutathione, so we do expect a certain level of cytotoxicity, but we also hope, based on the single cell analysis of DNA fission immunofluorescence that I showed before, we hope that those cells that are infected will die more preferentially. And this is what we saw when using the combination. So there was a level of cytotoxicity for uninfected cells, but the level of cytotoxicity for infected cells was higher. The final piece of in vitro evidence that we got here was that the frequency of integrated HIV DNA as measured by allo-PCR in cells of people living with HIV was lowered by the drug combination. And another interesting reason why this drug combination or the combined inhibitions of these two pathways might be promising is that it is feasible in vivo, although at this stage we only have a proof of concept piece of evidence on macaques. And I'm showing you only the two macaques that were treated with the full treatment. There were others that were treated only with ART or single treatments. But basically what was interesting is that after suspension of all treatments, there were several months of partial control of viral load that were observed. And the set point of the viral load was clearly lower in both macaques as compared to the pre-therapy set point. So, although I think we are the only ones so far who have tested the combined inhibition of these two pathways for HIV purposes, it has been tested in other disease indications, there is, I think, growing evidence that the use of oranophine could be an interesting candidate to try to reactivate latent HIV and potentially also reduce HIV reservoir. So in this study from 2021, the authors reported similar effects with only oranophine, in this case on JLAT models of latency. And in this more recent study, the authors studied PBMCs of people living with HIV, and they measured intact HIV DNA. So they did not see a significant effect, but they did see a reduction of intact HIV DNA. I think it's not entirely surprising that the effect was not significant, because oranophine per se is not going to induce a functional cure. It needs something else, perhaps BSO, as we saw in macaques, in order to be strong enough to induce a functional cure. And another layer of evidence that indicates that oranophine per se is not going to be sufficient is a recent clinical trial that has been completed at the Federal University of Sao Paulo, directed by Ricardo Diaz, and to which we have collaborated. And in this clinical trial, the post-therapy viral loads indicated that all patients receiving ART plus oranophine rebounded. Now, the multi-intervention arm that I'm not going to discuss at length did show some more interesting patterns of viral load rebound. So there are a few potential solutions to the fact that oranophine per se is not going to induce a functional cure. Now, one potential solution is to give BSO, as we did in macaques, and this is something that we're trying to do. So a clinical trial with a low dose of BSO plus oranophine, since oranophine has already been tested in this clinical trial in Sao Paulo and is safe for use in people living with HIV. Another option, which was incidentally what was tested in the arm, in the multi-treatment arm in the clinical trial in Sao Paulo, is to try to boost the kill stage. So try to use something else, not another inhibitor of antioxidant pathways, so that we can eliminate infected cells. And for that trial, it was personalized immunotherapy. The third option is whether we can find some alternatives to oranophine, and I would like to devote the last part of my talk to this and show you some yet unpublished data. So in order to explain how we came to the conclusion of which drug we might test, unfortunately, I need a second detour, I hope not too long. And this detour regards the promyelocytic leukemia protein. Now, the promyelocytic leukemia protein is one of the core components of nuclear architecture because it's the scaffold, the main scaffold of these PML nuclear bodies. PML nuclear bodies have a lot of different functions, but basically they were identified, PML was identified initially because of its link to cancer, its link to acute promyelocytic leukemia. It was also seen that PML can influence redox regulation and interact with NRF2. Moreover, the levels of PML have been linked to HIV replication and reactivation, as I will show. So one of the lines of evidence was, again, using the model that I showed towards the beginning of this presentation, where we looked that the number of PML nuclear bodies were higher in the cells after HIV replication went down or had ceased. Moreover, if we induced in cell line reactivation of the virus, we saw the levels of PML going down. So the higher the levels of PML, the higher the level of latency that we saw. In terms of its cancer connection, the reason why PML is important for acute promyelocytic leukemia is because the fusion product caused by the translocation between these two chromosomes is what causes the leukemia itself, which is basically a lack of differentiation of myeloid cells. Which brings me also to the first therapeutic compound that I want to discuss, arsenic trioxide. So arsenic trioxide is a really great drug for acute promyelocytic leukemia. And the reason it works is that it depletes PML. It induces depletion of PML through its sumoylation, ubiquitination, and proteasomal degradation. What is also important, however, and this is where the link with oranophine comes, is that in addition to the effect on degrading PML, arsenic trioxide can also inhibit thyroidoxin reductase, just like oranophine. So it does have a function against thyroidoxin reductase, but in addition to that, it has a function on PML. And other groups had shown that arsenic trioxide can reactivate HIV expression in cell lines and also decrease the amount of SIV DNA in macaques treated with antiretroviral therapy plus arsenic trioxide. So all in all, arsenic trioxide is really a great drug, almost 100% efficacy against acute promyelocytic leukemia. It's one of the tumors where the prognosis has shifted more dramatically, really. It's a candidate therapeutic for HIV for the data that I showed before, and is approved for human use. There are some problems with its use, though, and the main problem is it's an intravenous drug. It's costly and can be toxic, obviously, as the name also says. It contains arsenic. So the purpose of the study that we're currently pursuing is, can we come up with a cheaper, orally available alternative to arsenic trioxide, which retains the activity against acute promyelocytic leukemia, but also retains what we saw are the properties of arsenic trioxide against HIV. And the candidate that we are currently studying is Redox. And it's not something that we came up out of the blue. It has been used for millennia in traditional Chinese medicine. And a recent clinical trial, a phase 3 clinical trial, has shown that the use of Redox is as effective as arsenic trioxide for the purpose of treating acute promyelocytic leukemia. Another interesting line of evidence is that the lethal dose of Redox is higher, which means that the therapeutic window might be bigger for using Redox as compared to arsenic trioxide. So why does Redox not already be, why is not Redox yet the substitute for arsenic trioxide? The main reason is it's really poorly soluble. So the current protocols for its solubilization are very complex and pretty expensive. And moreover, its effects on HIV, to my knowledge, had never been tested before. So the first thing we did was to try to come up with a solubilization protocol that could be simpler. So it's based on pH and temperature. Now, to be honest, this solubilization protocol is a modification and hopefully an improvement over a very old protocol by Berzelius, which I think had been forgotten. It was published in the 19th century, so probably that's why people didn't really give too much attention to it. But as you can see, the eventual product of this combination of basically incubations in acid and alkaline solutions and final incubation at 65 degrees is a colorless solution. And this is the colorless solution that we use to test its biological properties. And we compare them obviously to arsenic trioxide as the gold standard. So the first thing we tried to do was to see whether the solution would retain the ability to deplete the PML protein in the tumor. That's the main indication of these arsenic-containing compounds. And indeed, this was the case. So Realgar did preserve dose-dependent effect on PML depletion, just like arsenic trioxide. So we were encouraged that we had not lost biologic activity through our solubilization process. The second thing we wanted to see was the transcriptomic effects. We wanted to see whether we could achieve a more targeted effect of Realgar, since, as I mentioned, its lethal dose has been reported to be higher. So in theory, it should have less also off-target effects, really. And what we notice is that on APL cells, the transcriptomic effects of arsenic trioxide and Realgar were similar. So we're talking about a tumor that is the target of these drugs. However, on Jurkat cells, on a lymphoid cell type, the transcriptomic effects of Realgar were definitely less visible than those of arsenic trioxide, suggesting a more targeted effect. So for our HIV work, we wanted to make sure, however, that Realgar did have the ability to deplete PML in lymphoid cells. So it's nice that it doesn't have a lot of off-target transcriptomic effects, but we wanted to make sure that we can achieve PML depletion. And indeed, we did see that Realgar was able to deplete PML both in activated and in resting lymphocytes. It was also able to decrease the number of PML nuclear bodies, which are really the structures that PML is forming the scaffold for in the nucleus. So the final thing we did was to try whether we could achieve some level of reactivation of HIV latency using this solution of Realgar that I just described in cells of people living with HIV. And what we saw was indeed that we achieved higher levels of HIV RNA in cells of people living with HIV, which was dose dependent when incubating these cells with Realgar. So to conclude my talk, I hope I could convince you that the antioxidant NRF2 mediated antioxidant pathways are important for HIV latency, and they seem to favor both the establishment and maintenance of HIV latency. We have identified glutathione and thyrodoxine as potential therapeutic target for trying to induce reactivation of HIV from latency, and also possibly partial selective death of infected cells. The nice thing about these two pathways is that there are good drugs to inhibit them both, and they can be used in vivo. However, there is also the opportunity perhaps to improve over one of these drugs, auranofin, by using an arsenic derivative which might retain some of the properties of auranofin while offering also the chance, for example, for dual treatments based on its activity on the promyelocytic leukemia protein. And I would like to conclude just by acknowledging my lab team and also the main collaborators that we're working with for our current work on Realgar. And thank you very much for your attention. Unfortunately, I didn't go through the chat during my talk, but I'll be happy to take any questions you might have, really.
Thanks, Luca. Actually, chat is mostly about the comment about you are not frozen. It looks like a technical side on my end, so sorry about interrupting your talk.
Okay.
Yeah. So so any questions, please? Amanda, go ahead.
Oh, thank you so much. What a fantastic talk and story. Congratulations on the work. I wanted to know, have you thought about the long-term effects of the potential treatment, right? You're blocking these antioxidant pathways. You told us very nicely in the beginning, right, how there's this balance. So I'm just thinking about, right, that, you know, what are the long-term possible impacts, and how could we get around those? Obviously, I'm thinking it could be damaging.
Yeah.
Thank you.
Thank you very much. It's a nice question, obviously. And yes, we did So regarding the inhibition of thyroidoxin, I don't think there can be a severe long-term effects simply because that drug has been used for rheumatoid arthritis, which means chronic use. So I would say it's reasonably safe to imagine a limited time of treatment for the purpose of trying to induce a functional cure of HIV. It is true that it's tricky to decide to inhibit both the thyroidoxin and the glutathione pathway in vivo. This has been done in mice for cancer treatment, and also we did it in macaques. But that's something that has to be carefully considered, and that's why in the clinical trial that we're trying to propose, we're starting from a dose of butyramine sulfoxamine that is very low. It's also, I think, worth noting that butyramine sulfoxamine would be given only for 48 hours. So it's really a booster given for a very short period of time, trying to eliminate as much of the reservoir as possible, while obviously causing as limited side effects as feasible. Yeah.
So perhaps then altering with regular art therapy.
Yeah, it will be. So this is given in any case in conjunction with ART, because you don't want to have a reactivation, obviously, in new infections, but it's given for a very short time window. I'm talking here about butyramine sulfoxamine, and then the idea is to stop all treatments and see whether there is a permanent functional cure, or obviously whether there is a rebound of viral loads. Yeah.
And very beautiful fish. So that was that first slide. That was a single proviral integrant because it was a single.
Yes,
is that right?
Yeah, that's a single. Yeah.
I've been out of the loop, but
yeah.
I wrote down, oh, I need to do this.
Yeah.
Thank you so much.
Thank you.
George.
Thank you very much.
Ron, go ahead.
Yeah. Thanks very much. I learned a great deal from that. And I was interested in having you talk a little bit more about effects on myeloid cells and the impact on the brain, because I think your talk was mostly focused on peripheral effects on lymphocytes.
So thank you. That's a good question, and apologies because I know that obviously the main interest of this group lies in neuroaids or the brain in general. So unfortunately, my own knowledge of the brain reservoir is very limited. That's why also we collaborated with the lab of Jonathan Karn to try to do some experiments on microglia. I have limited experience working directly on that. However, what I can say is that we saw robust reactivation from latency effects in this microglia model, and we did see also selective elimination of infected microglia. Whether none of the two drugs that we're using in normal conditions is expected to cross the blood-brain barrier. So we're not expecting to have also, that's a good thing, because obviously one of the problems of causing therapeutically oxidative stress is that you can lead to problems in the CNS. So I don't think there would be actually a penetration of these drugs based on studies on healthy individuals. But other than that, I'm really not an expert on that. Unfortunately, I worked mostly on peripheral reservoirs for most of my career. So,
yeah, there might be some effects because of the breakdown of the blood-brain barrier in people with HIV. And I'm surprised more of our myeloid experts aren't asking questions about this stuff, because there are several of them on the call. Not to put anybody on the spot.
Any myeloid people questions? All right. So look, I have one question. So you mentioned in the very beginning the role of keep NRF2, which is something that the medicinal chemists are really very actively working on. Mostly, we are trying to find something to liberate NRF2. And in your case, you like to really just downregulate percent. Is there any medicinal chemistry efforts on trying to really suppress NRF2 release? That might be something that.
Not that I'm aware of, because I think it's just, it would be too broad. So you would be, I mean, it's viable in cells if you knock it down temporarily, but it's not something that you can really pursue, because you would be basically stopping all or most antiviral factors from being transcribed and transmitted, and that would not be really something
I
that
see. So it probably is not a good pharmacological approach from a safety standpoint.
Yeah, you want to obviously induce some stress, but not to generalize
Exactly. Okay.
and block all the anti-oxidant response.
Sounds good. All right. Marcus, go ahead.
Yeah, thanks, Luca. It is really interesting, following up on what Ron said. I was also, I mean, hearing, that you want to increase oxidative stress, sounds a little bit scary if you think about a lot of the other organs, right? But you mentioned, I mean, there's also been clinical trials, right? And there is, the question is also, what do we know about the distribution of the drugs, right? Because, I mean, you wonder, I mean, certain drugs do tend, to accumulate more in some cells than in others. I mean, you showed some of the data, right? That with the with Jonathan Carnes or David Caball's microglia model, that you can actually eliminate some of the cells. But it seems compared, you had some really good effects on the lymphocyte cell line, but the microglia are a little bit less responsive there. But coming back, so what is known? You mentioned that some of the drugs are also used in rheumatoid arthritis, right? Which is a pretty bad situation, right? And there is in rheumatoid arthritis a lot of drugs are used because nothing else helps, right? And so, but has anyone looked at effects on the other cells, or are you planning to look at this? So, and what does it mean for the immune response in general? You get reactivation, but you have this oxidative. So how, I mean, what you would even think, how long do a lot of the cells expire faster because, even the periphery, because you have this, much more pronounced oxidative stress, what does it mean for the hematopoietic system, for example, right? I mean,
yep. So those are very good points. The metabolism of these drugs and the excretion and so on and accumulation have been studied. Regarding the immune function, that's a very interesting point because auranofin, I did not show it, but one of the reasons why auranofin is supposed to ameliorate, to improve the condition of rheumatoid arthritis patients is that it seems to target selectively the memory T cells. So there is a selective loss of memory T cells. And this is one of the reasons why, indeed, it could also lead to further differentiation of these cells, turning cells with a longer lifespan to cells with a shorter lifespan, which helps for the HIV reservoir, but obviously could have also other implications, for example, in the loss of some of the immune memory that was present beforehand. In terms of methionine sulfoxamine, so interestingly, the drug was initially studied in the 90s and early 2000s against glioblastoma. So I again did not mention that because it was not particularly in the focus of the talk, but it was used because melphalan, one of the compounds that was tested against glioblastoma, is detoxified through glutathione. So the idea was to increase the activity of melphalan with methionine sulfoxamine. Now, in practice, when the two were given in combination to these patients, I think the therapeutic efficacy was not great, and there were also 1 or 2 cases of severe toxicity. But when methionine sulfoxamine was given per se, so the depletion of glutathione was studied in several cell compartments, it was pretty generalized, but it was also quite temporary. So after, I think, 72 hours or something, people would go back to the normal levels of glutathione, which is, I think, also why the macaques did not display any particular toxicity out of that, simply because the treatment is quite short-term. It depends what you combine it with, because glutathione detoxifies a lot of things. So, for example, if a person is already receiving other medications, then you might have a situation where you increase the toxicity of those medications as well by giving something that depletes glutathione.
Oh, okay. Yeah, I know. I think these are, I mean, you basically have like a short-term oxidative stress shock, right?
Yeah, that would be also, like, sorry.
Yeah, but which raises you might actually get like a kind of a rebound, of antioxidant response afterwards, right? I mean, and that would be interesting to see if you get an even more pronounced, antioxidant response afterwards, right? And
no, yeah, this is not something that we've looked at. I don't know if someone else has. That's a good point. It could happen. The idea is if you can tilt the balance of the reservoir strongly enough, so that then you have spontaneous control. We did not get eradication with that treatment in macaques. What we got was partial control of viral load, which was also associated with cell-mediated immunity. I think this elimination of some of the previous memory could also help in that case, because you will have formation of new memory upon the rebound. However, I do believe that, and also the clinical trial in Sao Paulo seems to indicate that you will need some sort of either therapeutic immunotherapy or something that gives long-term protection after you have brought the reservoir down enough, because you will not eradicate the virus simply by eliminating some of the infected cells.
Yeah, but you may just, I mean, the question is, I guess arises if there is a possibility, because you wonder if you can do several cycles of this, right? If you can, over time, reduce basically the reservoir, right? I mean, it looked like you had also some data suggesting that even after, stopping, for example, some, maybe from the macaque models, right? That there was some prolonged time of control, viral control, right?
Yeah, so you could imagine having repeated cycles. I think you cannot overdo it, obviously. So especially with oranofin, as I said, you can keep people for months under treatment with oranofin, and there's no particular side effect. But when you give the combined inhibition, since these systems are redundant, you really have to be fast. So I think the time, we selected 48 hours, which is the standard administration of BSO. It cannot be very long, but it might in cycles,
Okay, thanks. Very interesting.
All right, any other questions? All right, thanks, Luca, again for joining today from all the way from England. It was a great talk. And Caroline, can you tell us about, can you tell us about the upcoming event? I guess we have Christmas and New Year. So when are we going to have this?
Yes, we're off for the
happen?
next few weeks.
Okay.
So we're back on January 8th. And the talk will be Nina Zhao of UCSD, and she'll present a talk, Drug Analysis One-Stop Shop, a comprehensive spectral library for drug and metabolites developed with public metabolomics data, and it's going to be moderated by Ron Ellis.
All right.
Good.
Sounds good.
Awesome. And again, we still have slots for anybody or your trainees that are interested in speaking in the spring. Please, please, please let us know. I know since we, said it last time, we had 4 more people sign up, which is amazing. So please, please, please spread the word. And I hope everybody has an awesome holidays. And we look forward, Ron, to you moderating our session right after the first of the year.
And thanks again, Luca, for the fantastic talk.
Thank you all.
And,
Yeah.
yeah. Have a good
Fabulous.
day, everyone.
Bye-bye.
Bye-bye.
Bye.